All right, we're going to have Fei Zhang presenting about assembly and reconfiguration in DNA, tile, and origami systems.
Thanks to the Foresight team for organizing such amazing workshop. I'm really excited to be here, and it's really nice to see those familiar faces. And also, it's very exciting to know new people and learn their research works. So today, I will share some of our recent work in my lab about DNA self-assembly and reconfigurations. So we have been always imagining what it looked like for our everyday life in future, and how can we be healthier and live longer. Imagine this, one day you can get a delivery box in front of your door, and that's the nano robot you ordered online. And those are bags of powders. You can mix them in a cup of water in your kitchen, and they can self-assemble into nanorobot. After you drink that cup of water, and those nanorobots will get into your body, check your health, and even repair things that need to be done, and send a report on your cell phone directly. It will not be a science fiction story if we work together to deliver such two boxes. So in my lab, we have been working on using DNA and RNA to create like programmable materials. And we create those fundamental design rules, and we interact them with other functional materials like inorganic materials, polymers, dyes, and eventually we want to deliver certain functions in different systems. And today, I will focus on the very first project that is established in my lab. You can see that it's a new type of DNA nanostructure. And started from that particular project, we extended into different research directions, included DNA origami and RNA folding, RNA co-transcriptome folding in vivo, and hybrid materials, and eventually some cellular functions that can be conducted by DNA. So today I will just talk about the very first project. So inspired by the crossover, anti-parallel crossover tile and a T-junction tile, we combined those two structures to create a new series of different DNA tiles, because those DNA tiles are the structural foundations for DNA nanotechnology. It enables new construction and eventually enable new functions. Originally, the crossover is the key for many constructions, including DNA origami, because crossover bundles up DNA helixes to rigidify that structure. But it ends up with this parallel helix routing. In order to create the second direction of that helix, T-junction is one way, but originally this T-junction tile is too flexible, so it cannot be assembled in solution. It only can be assembled with the help of a surface or a substrate. So we combine those two tiles together, we create a series of different layers of structure. You can see they have different orientations and have a strut to help them. And first, we demonstrate their assembly in solution, leading to a linear ladder formation or 2D array assembly. You will see those reinforced vertical arm and horizontal arms will lead to large 2D assembly. Some of them will end up with tubes, like this one. We also tested this layered configurations. You can see by layering up those T-tiles or inserting the third vertical arm into the space between those two helixes will generate some assembly at micron size, some tube or 2D array. So those just demonstration the feasibility of those new type of tile. The most exciting part, we found that due to the robustness of this tile, we are able to extend this tile to form this Z tile, C tile. And you can see by properly changing the length of those two arms towards C tile or Z tile, we can control the assembly of a nanoring. And we can even power the reconfiguration between those rings, ring size, or splitting, reorganizing those rings by simply using strain displacement reactions. And here is a simple demonstration to show how we change the size of each arms to be able to change the geometry of the individual tile and leading to the overall resizing of those nanorings. Here are 4 examples of showing the enlargement and the shrinkage of a nanoring with 8 monomers and 9 monomers, 12 monomers, and 14 monomers. You can see those ring has a diameter ranging from 36 nanometer to 94 nanometers. And to understand the resizing process, we conduct AFM image directly on the intermediate structures. You will see during the reconfiguration process, this intermediate structure remains the structural integrity of it. It indeed is a resized process rather than disassemble and reassemble process. As I mentioned before, this structure also can have 4-state switching or reconfiguration behaviors. What we have is if we start from a large ring like here with 25 monomers, and if we change the geometry of individual of this tile and lead to the new curvature of assembly, one large ring will be able to be splitted into several smaller rings. And you can see here we have 4 different states here, and we demonstrate all those reversibly reconfiguration, and in total we have 12 pathways. And you can see we demonstrate all these 12 pathways of reconfiguration, but the bad news is not every of them have high yield. So generally trend, if the ring is smaller, containing smaller amount of monomers, it generally has high yield. But if a larger ring, for example, for the first one, the formation yield is relatively low. And the most interesting part is how those large nanorings split into smaller rings, right? We apply the same technique by directly image those intermediate structures at different time spots, and after adding the invader strand at 1 minute, 5 minutes, until like 2 hours, we found that just at 1 minute, most of these large rings, which is 8 monomer ring, will pop open into a curved ladder. And at 5 minutes, smaller rings start to show up. We are lucky to catch some intermediate structures, as I highlighted here. You will see this curved ladder adopt half of this original 8 monomer ring curvature at the right side, and the new curvature for the target smaller rings. So basically, after we supply the invader strand, the large ring popped open, forms a curved ladder, and it will start to adopt the target shape. Eventually, they will form different smaller rings. But you can see this strategy is limited to like roughly control the ring size. We always have a yield. To better program those nanoring assembly and have a fully addressable surface, we also adopt a single-stranded tile strategy into our system. In this way, this 8 monomer ring, the geometry of it will be not just controlled by the individual of the tile, it's also controlled by the matching rules between all those tiles. So we demonstrated those ring assembly, and we found that this smaller ring will have a higher assembly yield, up to like 76%. And it also demonstrates similar reconfiguration process with this curved ladder intermediate structures. So to wrap it up, compare with our nanoring reconfiguration from DNA tiles with DNA origami, right? Here is like a work we just published last year with our co-workers. We designed a DNA origami with a reversible opening and closing lid that works as a nanopore. And compared with this design, it's statically designed, accurately controlled. But think about the cost. We need more than 200 staple strands with unique sequences. But for our DNA tile system, each of these individual monomer that lead to this large ring and reconfiguration, most of them, they need less than 5 DNA strands. So the reduction of the cost is dramatic. But we still appreciate the accuracy of DNA origami and the addressability of DNA origami. So at the last, I think it's safe to say that from the DNA and RNA design is a great example to show the information direct structure and eventually a function chain of this. Those DNA nanostructure are dynamic, self-replicating, adaptive materials, and they can be designed across length and time scales. And eventually, we want to use those nano devices to interface with living cells and to allow us to live healthier and longer life. That's all I have. Thank you for being here.
All right, questions?
Thank you. You mentioned a number of times having inputs to make these reconfiguration changes. Are those inputs always just other strands, or do you also change other parameters like temperature or pH?
That's a very good question. So, because strand displacement is very robust, all the dynamic structure in my lab, I always ask them test the strand displacement first, and then we order like a light responsive like a strand, and also pH responsive strand. So it's a peptide. I think many of the mechanisms have been demonstrated in the field already, yeah.
So you have a lot of options.
Yes, absolutely. Yeah.
Thanks, Fei. Really nice work. I was also, two quick questions. One, for the tile systems, are all the actuating strands the same sequence, or are those addressable kind of around the ring?
That's a very good question. So for the for the tile that with repeating unit, so that one use single input. And for the single strand tile with the 6 different sequence monomers, we use different sequences.
Okay.
So we demonstrated both. So they have advantages and disadvantages.
Yeah. And then second quick question, do you think the breaking that you're seeing in these intermediate states, do you think that's happening in solution, or do you think it could be some interaction with the surface that sort of triggers that breaking?
That's a really good question. Actually, we don't know, because AFM imaging is always something deposit on the surface, then you can image it. If we want to have some real-time monitoring of that process, probably I need to label certain location in that ring to be able to study the distance of points of interest.
Great. Thanks.
Thank you.
So now the question that we are asking everybody is like, how do you see the work integrating into a larger system?
I think for DNA, it has a lot of potential, since all the backbone and a different design, it just involves sequence. For example, those dynamic rings, maybe we can fix onto different origami framework. And for DNA origami, we have a different demonstration, like from William Shi. We have different scaling up method, to connect origamis together to reach a larger assembly, basically.
Thank you.
Thank you.